Our 24-hour man-made clock was invented and designed to mimic the Earth's internal clock. The Earth's rotation has been found to impress a 24-hour rhythm on the genes of all living things, from flowers to whales, even bacteria. All living things, it seemed at first, except humans. Multiple past studies measured human circadian rhythms to be a little over 25 hours. Thus, until more recently, it was believed that natural light and other cues had to reset our biological clocks over an hour each 24-hour day. This was a bit of a puzzle. Why would humans' circadian rhythms differ from those of all other known living things and make such a significant frequent reset necessary? This mystery was solved in 2016, however, by a leading researcher on circadian rhythms, Seisler. Seisler reported a series of studies we will call the measurement studies, in which he developed a new, more valid and reliable measurement system and found that humans' rhythms, like all other living things, are closer to 24 hours than 25. They are, in fact, 24 hours and 11 minutes. And even morning and night people vary only by 16 minutes in each direction. So human circadian rhythms are about 24 hours during which your behavior and physiology vary in predictable ways. In other words, behaviors such as your sleep, cognitive performance, and alertness function in predictable ways at predictable times within each 24-hour nycthymeron, as does your physiology, such as your body temperature, your hormones, even your urine production. This human clock consists of a cluster of nerve cells in the suprachiasmatic nucleus, barely 1/100th of an inch in size, located deep in the brain and connected to the eye's optic nerve. In fact, we saw evidence of the circadian rhythm on alertness when we viewed these MSLT sleepiness scores for a normal person during the day. Remember the afternoon sleepiness peak right here? Sometimes this is referred to as the afternoon sleepiness peak, at other times, the afternoon alertness dip. Opposite sides of the same coin, right? There are four major ways in which circadian rhythms are studied. With the first, entrained 24-hour protocol, participants live in a lab with exposure to natural lighting. In other words, a lab with windows, for a period of time on a typical 24-hour schedule of sleep, wake, meals, and activities. Biological and/or behavior measurements are taken regularly to assess their 24-hour rhythms under these normal circumstances. In the other three methods of studies, participants need to be removed from environmental cues, so they are conducted in sleep labs with no windows, no clocks, no media, no phones, etc. So let's look at the first, free-run experiments. Participants have control over artificial lighting, and they determine when they want to sleep, eat, or engage in activities. So they do whatever they want, and their sleep-wake patterns are recorded for weeks, while participants rely on their own internal clocks without any outside cues. In contrast, forced desynchrony studies require participants to try to follow a very abnormal and pretty impossible schedule involving bed and darkness for the first 1/3 of a cycle, followed by spending 2/3 of it out of bed in the light. Typical cycle studies are 20 minutes, 90 minutes, or 28 hours. Imagine being a participant in, say, a 20-minute cycle study. You would agree to go to bed for 7 minutes in the dark, then be up for 13 in the light, then repeat this over and over for usually 24 hours. Researchers would be interested in measuring the amount of sleep you would be able to get when your sleep-wake cycle is desynchronized, in other words, dramatically cut off from your circadian cycle. Finally, constant routine studies require that participants are in a semi-recumbent position in a quiet, dimly lit environment, but are kept awake the entire time with constant psychometric tests. Small meals are eaten every hour. Constant routine studies typically last over 24 hours, but never over 48.